Now we're going to talk about nucleic acids, and apologies in advance, there will be no funny cat noises because I'm recording this at school. So let's talk about nucleic acids. So the monomer of nucleic acids are titled nucleotides, and they have three specific parts that make them up. The first part is a 5-carbon sugar, and I did point out before that a 5-carbon sugar is called a pentose. So if I use those terms interchangeably, hopefully it's not too confusing. There is also a phosphate group, and we're going to talk about that. I'm going to make a note about that in a few slides. And then there's also a nitrogenous base, and that nitrogenous base, there's a couple of different ones, and we'll talk about those on a future slide as well. So these are the three components of a nucleotide. Every nucleotide has at least those three parts. So let's talk about the 5-carbon sugar. The pentose in DNA is going to be a sugar called deoxyribose. And remember, when you see ose at the end, that does mean that it is a sugar. And then the 5-carbon sugar in RNA is going to be ribose. So that's actually where the names of deoxyribonucleic acid and ribonucleic acid come from, literally from the sugar that is a part of the nucleotide. You do not need to know the structural differences between those. You just need to know the identities and the fact that they're found in different nucleotides. So you'll have deoxyribose found in all DNA nucleotides and ribose found in all RNA nucleotides. Let's talk about the nitrogenous bases. So nitrogenous bases can be split into two different categories. Pyrimidines, which are pictured on the top of this diagram. Notice what they all have in common is they all have one carbon ring or one hexagon making them up. And then the identities of them are cytosine, thymine, and uracil are all examples of pyrimidines. Then you have the purines. Notice that they have two ring structures, a pentagon and a hexagon. And I know I didn't actually draw those, but and those have two rings, so those are adenine and guanine. Now, for the actual AP test, you don't need to be able to tell which nitrogenous base is going to be a purine and which one's going to be a pyrimidine, and it's very unlikely that you'll need to be able to identify a purine versus a pyrimidine based on how they look. But when you're doing college level biology, it is possible you'll have to memorize a little more about their structure. So I just wanted to give you a tip for that in the future. One of my former coworkers who went to A&M said that she was given a hint to help her remember which of the nitrogenous bases are purines and which are pyrimidines, and she was told to remember Aggies are pure. Keep in mind she went to A&M, but that is because the A and the G, the adenine and the guanine, are purines. I also remember which of these is which by remembering that the longer word goes with the shorter molecule. So pyrimidine, long word, shorter molecule, it's only one ring. And then purine is a shorter word, but it goes with the longer molecule. And if that helps you in the future, hopefully you can remember that. All right, a very brief note about nucleotides. So nucleotides, you may remember learning about ATP. ATP is essential. We're going to talk about it in more detail, and it is used for energy transfer. And you may have thought, oh, ATP is something different. It's special to have those three phosphates. You may also remember learning about ADP, adenosine diphosphate, which is the form of ATP that has less energy. But we're going to talk this year also about some monophosphates. So we're going to talk about AMP, or adenosine monophosphate. And one thing I wanted to point out is that in the cell, naturally occurring, most of these nucleotides actually exist as triphosphates. They're going to be, there's ATP, there's CTP, there's GTP, there's TTP, and there's UTP. So you'll have a uracil that's going to have 2 extra phosphates on it, a guanine that has 2 extra phosphates on it, and then those extra phosphates are cut off during the process of dehydration synthesis to make a polymer and make a larger nucleic acid. But there are 2 of these nucleotide triphosphates that are important. So you know about ATP, adenosine triphosphate, and we're also going to learn about guanosine triphosphate, or GTP, when we talk about cell signaling. So I just wanted to make sure that you guys had this note. And then also just to note that if you've got a nucleotide with 1 phosphate attached to it, then it's a monophosphate. with 2 phosphates attached to it, it's a diphosphate. And then just like with ATP, the 3 phosphates attached to it make it a triphosphate. So all nucleic acids have directionality, meaning that they have a firmly, just like when we're writing words, we have a left side of the page and a right side of the page, and you always write from the left to the right side. With DNA and RNA, you always they have specific distinct sides, and you always build them by adding to only one side of it, and you actually read them in a certain direction as well, which we'll talk more about when we talk about genetics. So if you look at the pentose, at the 5-carbon sugar, so this is going to be our sugar here, and here would be a nitrogenous base, and then you've got phosphates coming off there. We're not going to go into why this is the case, but you've got an end of the sugar called the 5' end and an end of the sugar called the 3' end. So when you're looking at nucleic acids and you're building them, they are always built in the 5' to the 3' direction. So you always add to the 3' end. And the way I remember which end is which is I think that the F in 5' sounds like the F sound in phosphate. So the 5' is the phosphate end and the other one is the 3' end. You are not going to have to be able to recognize these on a diagram. They're never going to just stick a diagram of a sugar and ask you to say, okay, which is the 5' end and which is the 3' end. What they'll usually have is on their diagrams of DNA or RNA, they'll label the 5' end and the 3' end for you. All right, and so this is also important to note because, like I said, there's a 5' end and a 3' end. When you have double-stranded nucleic acids like your DNA is said to be anti-parallel. So the 2 strands of DNA are parallel, meaning they run, parallel to each other, but they run in opposite directions. So you'll notice on this top strand, it runs 5' to 3' from right to left, and the bottom strand runs in the opposite direction from left to right. And there is actually such a thing as double-stranded RNA. RNA can form some double-stranded sections. It's usually not the entire molecule, but where it does, those areas are anti-parallel as well. So hopefully you remember from freshman biology, there are base pairing rules. What we didn't talk about in a lot of detail is the fact that these base pairing rules are determined by hydrogen bonding. So hydrogen bonding happens between the 2 nitrogen bases. Remember I said when you have any NH bonds, those tend to be polar, which means they're capable of forming nitrogen bonds. So the nitrogen bases will form hydrogen bonds with each other, and that's what holds double-stranded sections of nucleic acids together. So in all nucleic acids, guanine and cytosine will bond with each other and they form 3 hydrogen bonds, like what you see here. And then hopefully you remember that you form different bonds with adenine depending on which of the molecules you're talking about, but adenine always forms 2 hydrogen bonds with whatever it's pairing with. The difference is that while cytosine always pairs with guanine, adenine pairs with something different in DNA. It pairs with thymine, but there is no such thing as thymine in RNA, so instead it pairs with uracil. And we'll go over that more when we talk about genetics later. Some of the structural differences between DNA and RNA are what the 5-carbon sugar is. So remember, DNA contains deoxyribose, RNA contains ribose. Then all of the nucleic acids have 3 nitrogen bases in common. They have adenine, cytosine, and guanine. But in DNA, you have thymine, and in RNA, those thymines excuse me, are instead replaced with uracil. Another thing, DNA is always double-stranded, whereas by default in general, RNA is usually single-stranded, and we'll talk about some exceptions to that later. Okay, And we're going to focus more on the function of these nucleic acids when we get to Unit 6 and talk about genetics. For now, we're mostly focusing on the structural differences. And then here is your summary page. So nucleic acids in general, they store and transmit genetic information, which is ultimately going to be instructions for creating proteins. DNA is always located in the nucleus unless something has gone wrong, which we'll talk about later. And RNA can be found in the nucleus and in the cytoplasm. The monomer is a nucleotide, and the most important examples of nucleic acids are going to be DNA, RNA, and ATP, which in freshman biology we don't tend to emphasize as much that that's a nucleic acid, but you do need to know it here. And that is the end of this set of notes.